Just thank you very much for having me. I'm coming from a genomic evolution lab, so I'm not very in my fish tank because I normally go to evolution meetings, but here I am. And it's because I want to show what we as a genome evolution lab are doing with many of the tools you have developed and some of the tools that we have developed. And this is mainly the work of a very talented postdoc that I had the chance to supervise, Quentin Rogemont, and that he's presenting in an evolution meeting, the exact same work. So and a bunch of other people that were involved. And so rather than telling you what we did, I'm going to tell why we did it and how we ended up doing it. This is more like my approach here. And so why we did it is because we're interested in when recombination stops, and this kind of an evolutionary puzzle, because recombination is great in the sense for the diploid organisms. It's always secure that one of the meiotic products are going to have kind of the best collection of alleles. It's just for sure this is going to be, even if they are full of deleterious mutations, one of the meiotic products is going to have a better contribution to the next generation. So that's great. But sometimes recombination is not, it's selected against when you have the case in which a locus needs to be in a permanent heterozygous state. And so when this is a necessity, so the selection will act to prevent recombination so that recombinants are never, are less fit than non-recombinants. And this is actually the case. I'm going to skip this one. The case of sex chromosomes, that's the most commonly known non-recombining chromosomes. That's what I asked, the question I asked before. And in one of the kind of interesting facts about sex chromosomes is that once they stop recombining, the recombination kind of grows around. And it doesn't, and when recombination suppression grows around the non-recombining loci, and when it does that, then since you are not recombining, you cannot kind of fix any kind of deleterious alleles that are being accumulated, whether by sequence divergence or invasion of transposable elements or whatsoever. And so you have this kind of process that it's in the end is deleterious. And we have even cases in which the heterogametic sex chromosomes has completely disappeared in some mammals and in many insects and some other systems. And a kind of interesting pattern is that this recombination suppression do not extend gradually, as if just recombination, like some kind of linkage disequilibrium with the non-recombining zone locus, it's just, there are less recombinations, so just by chance you kind of grow little by little. No, it happens like blockwise. A single block entire stops to recombine in this kind of blockwise manner or stepwise manner. We call that evolutionary strata, and it's a landmark of sex chromosomes. And how do we see this evolutionary strata is that we plot a synonymous divergence between the two haploids that stop recombining, and you see this kind of pattern in which you have so the original recombination suppression, high synonymous divergence, then the next block a little bit less, the next block a little bit less, and so on and so forth. We humans, we have like. And so what we thought is that now that we are able to sequence, to have haploid resolve assemblies, we may use these kind of beautiful tools that people like you had developed just to put them all together, that we can go just from genomes to infer this kind of blockwise divergence along ancestral synteny blocks. And before I got this question, when I say ancestral synteny blocks, I'm referring to the recombinant state as if it happened before recombination was suppressed. And so we had applied this tool for a bunch of systems I'm going to tell you a little bit about. But first, I would like to do why we were kind of forced the way to do it the way we did it. And so this is maybe I haven't seen this particular plot, and in other conferences this is a very common one. It's just number of genomes, and as the genome revolution has just exploded, so we have more and more and more genomes. But this explosion of genomes has not been accompanied, at least not by an explosion of annotated genomes. So we kind of are stalled. At the beginning, we had tons of genomes, from almost every single new genome was annotated, but then we start sequencing, you and me, we start sequencing like crazy, and genome annotation just stalled. And now we are more or less around 75% of all the species, and not all the genomes. I'm just putting one species, at least one genome per species, and at least one annotation per genome. And the other problem, so we have, since our inference is based on synonymous divergence that we can calculate only in coding sequences, so not having annotated genes, it was a problem. And the other problem is the problem that sex chromosomes are not recombining regions, so they tend to get scrambled. And so if you want to see a nice diagonal like that in this kind of dot plot, it means the two haplotypes are completely collinear. That's perfectly perfectly reasonable. But as soon as you go to the non-recombining part, there's only tiny little pieces that are still recombining, and the rest is completely scrambled. And this is partially because these regions had accumulated a lot of repetitive elements, and they have this kind of non-homologous recombination between them, and so they just scramble. And if you were to calculate the synonymous divergence along this kind of order, so you don't have access to this ancestral synteny, like ancestral genome order, so there is no blockwise. It's just like every single gene is just going in a different kind of selection process, and then you say, these guys, they had accumulate a lot of, so they may be doing something interesting. No, let me stay with me. And so what Cantan did is that he put all the different pieces of custom-made code and very environment-specific pipelines that I had for genome annotation, synteny determination, synonymous divergence calculation, and then kind of this kind of ancestral state-based strata inference in a single workflow that takes a genome and gives you evolutionary strata. And so there are 4 modules. The first is just a genome annotation. People would like to use rather the next flow alternative. This is the way we do it. We did it. Either we just take the genomes, we qualify the genomes, then we ask, do we have RNA-seq, so to inform our gene predictors. And if it is the case, then we map we map the reads, and we call genes with breaker and zebra. And if not, we are going to go and search if there is some related species that we trust the annotation. We can inform as well the ab initio gene modelers. And if not, then we are going to go and take OrthoDB or whatever. So we are very generous, so we're going to provide everything for you. So we are going to provide the mapping, and the annotated genome as a GFF or as proteins and CDS that you can keep playing with our tool. And our tool in this case is that we are going to take now these newly predicted gene models in a single framework. We are going to feed them to GeneSpace, and so GeneSpace are going to run OrthoFinder or MCC scan behind the curtains. We are going to provide all the outputs of these guys. Then you will have these kind of beautiful riparian plots that for me are not so easy to read. But when you have these riparian plots or these orders of this gene-based synteny, then you can make a circus that some people are very prompt or they like it a lot, or just regular dot plots. I'm a very old bioinformatician, so I'm very used to those ones, and this is the way I understand them better. But you are going to have them all, and the and the raw output of gene space, plots, but also analysis, including single copy genes in which you can calculate synonymous divergence. And that is what we're going to do next. We are going to have a codon native aligner that is called Maxi, feed them into PAML, forget about struggling with the single control file for every single CodeML run that you have. We are going to do it for you. And we are going to provide a separate folder for every single of the alignments. This can be in the numbers of the thousands, but it's going to be everything in a single folder for you. And so I'm going to take just a little detour and to say this is the last part. So we were very happy. We were applying these to fungal genomes. They are very small. They don't pose many problems actually to annotate. And so we were invited to participate in this huge consortium that is the White Campion, the Silene latifolia Y chromosome consortium. That's more than 50 people. And so we start participating to this, and some people noticed that we had kind of developed this metric to try to pinpoint exactly the place in which these kind of different blocks appear. And we had this very naive approach, which we're going to have like average over windows, and then we are going to subtract the average of one window to the next, and the one window to the next, and the one window to the next. And we believe when the difference is the highest or the lowest, so the difference is in absolute values the highest, that will be the boundary of the strata. And then Deborah Charlesworth said, that was part of the consensus, say, but you may be reinventing the wheel, because these guys is something that is very well known in the stock market. This is trend analysis, and there is something I think is called change point analysis, and you may want to try it. And actually, for sure, we did it. I mean, if Deborah says you do it, and so it worked marvelously. So we have been able. So the little these things on the bottom are the chances of a change of train to happen exactly there. And I am not, this is not my data that I know very well by heart. This is just fish that we took to prove that this is working for anything. We already proved with the Silene latifolia genome, but we wanted, so fungal plants, but maybe animals are different. So we did it with a fish, and we reproduced almost to the base pair the things that they had reported several years ago. And so we were very happy. We submit this for publication. It's under review right now. Should be okay soon. That's the GitHub site that you can clone and run it by yourself. So what we have done with this, and what we believe it can do more than that, is first, it can do all the different modules. If you are not interested in evolutionary strata but are struggling with CodeML, just feed them the alignments and it's going to work for you. There are many different options. So Cantan has prepared a very extensive documentation with many examples. But what we think is more interesting here is that this is not just sex chromosome specific, but it can do whatever it's in whatever process in which recombination has not occurred the same way all along the genome. And so that we just heard yesterday, genomes are not just a witness what they do, but also how they had become to do what they do. And so so anything where recombination behaves or has decay blockwise, then this is your go for to. Just finishing, there is some other signals of this process of how evolution is being recorded in genomes. And one of them, my favorite one, is what this kind of branching patterns that we call trees, even if they are sometimes networks. And something that it's kind of really obvious is that different parts of the genome tells different part of different stories, and these stories are sometimes related because of different selections processes or different population processes that came into play at different times. So I was kind of happy already to be able to identify the different regions, but then I wanted to also say how different they are. Can I develop something really new, not use things that were already there? And so I thought maybe I can compare the trees to one another and make not a Robinson false distance or whatever that is kind of, it's good, but it's not doesn't tell all the story. But thank you very much. So I'm gonna use this always or this approximate unbiased test that I think is a hidden gem in the field. No, not many people use it, but I think it's fantastic. It's just going to tell you whether a tree is coherent when with an alignment. And so your alignment is going to give you a maximum likelihood tree or a Bayesian best posterior tree, but it's not. There might be another one that is as good as this one. It's just not being output by the program. And so this test was developed to distinguish when trees are really different or not coherent with one. But I think I may just kind of repurpose this as a tree consistent metric. So I did it. It worked like a charm. When I put this is just Markov clustering on the approximate unbiased p-values of the trees. And as you see, though different colors are different evolutionary strata, when they form a cloud, a clique, that means even if not all of them are coherent with one another, it means they are more coherent within than outside. We just hear it out. And when you put that back on genomic coordinates, it looks like that. So actually, it's telling you which regions of the genome had followed kind of the same evolutionary story. And in this case, it's a kind of straightforward because we know this is the stepwise or block-wise extension of recombination suppression. But I think we can use it for other things like phylogenetic binning and so on. And so if you know anyone that would like to join me and to do that, I'm more than happy to talk. Or you yourself, I don't know. And for that being said, it's just, this is not only my work, it's mainly Cantant. All the people with arrows there are co-authors of this. But it would have not been possible with a couple of alumni, Sarah now in the States, Hélène in Toulouse, Fantan in Belgium, Marine in Germany. In particular, Fantan and Marine were my two former PhD students, and I'm very proud of them. And then all the external, in particular, Matteo and Aline, that were kind of the people that actually try our tool in their own stuff and actually kind of worked. So, of course, Deborah and Gabriel, who invited. Thank you very much. Happy to take questions.
All right, I think we can squeeze in a question at least. Yeah. All right, anything on anybody online just before we take over? All right, yeah.
I have a.
Thank you.
So another example of something where you have recombination suppression is meiotic drive systems,
Yes.
like the T haplotype in mouse. And so do those form strata? Have you tried applying it? Do you have a one sentence, two sentence sort of?
One sentence, meiotic drive, we had not succeeded so far. Supergenes, yes, beautifully. Butterflies, ants, that works very well. Thank you.
I'm waiting. I'm going to be on the edge of my chair.
Okay.
Do it.
Fantastic. Let's thank you one more time.